Welcome to the Galien Foundation Week of Innovation webinars here in New York. My name is Dr. Theodore F. I am the CEO of Skipta, Informa Pharma Intelligence, and have the pleasure of serving as the moderator for this Galien Foundation session today. Objective this week is to provide additional content and background for our largest annual event, the Galien USA Awards, to be held this Thursday here in New York, in conjunction with our annual Galien Forum of speakers representing top scientific experts and leaders in global health. Because the mission of Galien is innovation to improve the human condition, we have developed this new webinar series to give our nominees for Best Pharmaceutical, Best Biotech, and Best MedTech product greater visibility in describing their journey of medical discovery. This year's Galien USA Awards has a record 67 nominees for our three categories, from 54 companies covering 13 diverse therapeutic areas, ranging from cancer, cardiology, and neurosciences to infectious and very rare genetic diseases. We will be looking closely at the products in all these critical areas throughout the week. Here are the details of today's webinar. Because of the number of nominees and the range of conditions represented by all this excellent science, the Galien USA Awards Committee has decided to host a series of 10 webinars prior to Thursday's forum and award ceremony, where the winners in each of the three best of categories will be announced. The 10 webinars will drill down on the different therapeutic focus of the products, the quality of the science behind them, and most important, the patient clinical impact. It's intended to give our award attendees, the media, and the public a fuller picture of the innovation behind the medicine. In the best pharmaceutical category, which is our focus today, we will be holding 4 webinars due to the large number, 23 in total, of products representing this important field. Members of the awards committee decided to arrange each of the 4 webinars to center on specific therapeutic drug segments. These are chronic diseases, solid tumor cancers, hematologic cancers and other rare diseases, and infectious diseases and reproductive health. This webinar, in the hour we have allotted, is devoted to the 6 nominees in hematologic and other rare diseases. What we will cover today relates to this central question: are there common links in advancing research on hematologic cancers and other rare diseases associated with uncontrolled vascular system inflammation, such as sickle cell anemia? Researchers have found that patients with sickle cell disease have increased risk of developing blood-based cancers like leukemia. Some evidence also exists that the pediatric immune disorder, HLH, sometimes occurs simultaneously with lymphoma and acute myeloid leukemia, a situation that is often not diagnosed, leading to complications in treatment and the death of young patients. And now let's move right into our discussion. And it's absolutely my pleasure to welcome our panelists. Welcome panelists to our video today. Thank you so much for joining us. We really appreciate it. At this point, I'd like to introduce all of our panelists by name. First off, please welcome and say hello to Dr. Ramon Tu. Dr. Tu is the global medical leader of Astellas Pharmaceuticals Incorporated. Dr. Tu, welcome. Thank you.
Thank you.
Also joining Dr. Tu will be Dr. Michael Kaufman. Dr. Kaufman is the Chief Executive Officer and co-founder of Karyopharm Therapeutics. Dr. Kaufman, welcome and thank you for joining us today.
Pleasure to be here.
And our third panelist today is Dr. Mimi Huizinga. Dr. Huizinga is Vice President, Head of Oncology Medical for Novartis Pharmaceuticals. Dr. Huizinga, welcome and thank you so much for your time today.
Thank you for having me.
And to all of our panelists, again, thank you for your valuable time. We're really excited to spend the next 60 minutes with you. Just before we begin, a note to our audience that there is an accessible link to a 100-word statement submitted by the companies on each of the 8 nominated products. This link is available for viewing during this webcast. Also, we will have a poll that will now post up on the screen at this time. Galien Foundation, I'll let you post it. And hopefully that poll now posted to your screen. We do ask, we'll take a few moments here, if you could please complete this poll. At the end of our presentation and discussion today, we will post the results and review them with everyone participating. So thank you so much for participating in our poll. And thank you for that. And with that, we will begin our session. And our session today, I will ask our panelists a number of questions. And panelists, thank you so much for your time in answering these questions. We're really excited to hear the input and really the valuable research and innovation that you and your companies are really bringing to the world through science and innovation. So with that, I'd like to welcome again Dr. Ramon Tu. Dr. Tu, thank you again so much for joining us today. First question we'd love to get your input on is as follows. What did your company choose to focus on the disease state and indication associated with the product? In what ways did the product represent a therapeutic breakthrough? And how significant was the level of unmet medical need? Dr. Tu, I welcome you to the camera. And again, for our audience, if you wouldn't just remind them of your product, your company, and your background before you answer our question. Thank you.
Thank you so much, Theodore. And again, welcome to everybody who's part of this very important and exciting meeting. So as mentioned, my name is Ramon Tu. I represent Astellas Pharma. I am an executive medical director and currently serving as the global medical lead for our gilteritinib program. So without further ado, in terms of the specific question that was asked, so the focus in terms of gilteritinib as a product is primarily in a specific disease called acute myeloid leukemia. It is a type of hematologic cancer that certainly afflicts a large number of adult patients who develop acute leukemia. It is considered the most common type of acute leukemia in adults and primarily affects older patients. About 30% of these patients have a genetic mutation in a specific gene called FLT3, and that particular genetic mutation, if present, is associated with very high relapse rate and shortened overall survival. And it's important to, I think, put it into context because despite advances in chemotherapy that has occurred for quite some time, it is important to take note that little progress have really been made for AML as a whole. Can you believe that the progress as far as chemotherapy dates back to 1970s, and there's really been a paucity of new therapies that can improve outcomes for this disease. And that is really the major reason why Astellas decided that this is something that it would like to focus on. The unmet medical need in the area was clear, and Astellas was motivated to focus on innovation where limited treatment options are available, and where there is this greatest opportunity to help people who are living with these hard-to-treat cancers. That's why we aggressively pursued Zospata, also known as gilteritinib, and it was discovered through a research collaboration between Astellas as well as Kotobuki, and it was our first oncology compound identified and developed entirely in-house at Astellas. In this particular specific therapy, it is now currently approved for patients with relapse refractory FLT3 mutated AML.
Dr. Tu, thank you very much for your response. And I'd like to welcome Dr. Kaufman to the camera. And thank you again, sir, very much for joining us today. Same question to you, just to repeat for your benefit and for our audience. Why did your company choose to focus on the disease state and indication associated with the product? In what ways did the product represent a therapeutic breakthrough? And how significant was the level of unmet medical need? Dr. Kaufman, welcome. And again, if you could remind the audience of your product and your background. Thank you, sir.
Sure. Our product from Karyopharm is Dacvio. It's the brand name. This is selinexor, also known as KPT-330. It was our first compound coming out of Karyopharm Therapeutics. The company was founded by Dr. Sharon Shacham back in 2008, and I was a co-founder for that. We are a husband and wife team. She's currently the president and CSO of the company, and I'm the CEO of the company. I was trained as an MD PhD in internal medicine rheumatology. The focus was actually fairly broad, and it's panning out pretty well. Obviously, cancer remains a great unmet medical need, and I mean this in the sense of the many, many types of cancers there are. Most of these cancers are still not curable, and most patients will require additional therapies after frontline or second line therapy. What Dr. Shackham set out to do was to attack one of the pillars or the hallmarks of cancer that hadn't yet really been addressed. Things that we have done very well in cancer is take advantage of the fact that we know that cancers are driven, for example, by oncoproteins. Novartis has a beautiful product in Gleevec, the first in class that attacked one of the specific oncoproteins that drives a certain kind of leukemia. This was game-changing, and although not curative, it's a remarkable breakthrough drug that really revolutionized the treatment of chronic myelogenous leukemia and other leukemias. And similarly, we've heard about glofitinib and others. We've attacked oncogenes. We have attacked vascular disorders that are associated with cancer in the sense that we have drugs that hit VEGF and prevent the vascularization of solid tumors, and drugs like Avastin and related compounds have really changed solid tumor treatment for many people. But we have not really addressed one of the hallmarks, which is the fact that tumor suppressor proteins, that is the proteins that all of our cells have, there are about 20 of these, that are turned off when a cancer grows. And it's been somewhat of a mystery, actually, how a tumor cell manages to turn off 20 different proteins when only a small subset of these proteins are mutant or mutated or changed in the tumors. And what Dr. Shackham was able to glean from the literature is that one of the simplest ways to do that is to simply move the tumor suppressor proteins out of the cell nucleus and into the cytoplasm. And what that means is these tumor suppressors no longer function. Tumor suppressors have to be alongside the DNA in the cell nucleus in order to work and suppress tumor development. They work in most of us, but when we get cancer, they stop working. It turns out, for reasons that are only clear in evolution, and we don't understand them yet, that there is a single chaperone or carrier protein whose job it is to move tumor suppressors from the nucleus into the cytoplasm. And that protein is called exportin 1. It's one of 8 exportin proteins that act in our cells, but it's always the protein associated with moving the famous tumor suppressors like P53, the guardian of the genome, and BRCA1 and BRCA2 and retinoblastoma and so on out of the nucleus, and thereby inactivating them. XPO1 is overexpressed in essentially every tumor that's been looked at, from leukemias to myelomas to lymphomas to solid tumors, sarcomas, and so on. And blockade of XPO1 was not possible prior to the advent of XPOVIO. XPOVIO is the first and only selective inhibitor of nuclear export. It blocks exportin 1. It leads to accumulation of tumor suppressor proteins in the nucleus, which leads to selective death of tumor cells. It doesn't kill normal cells because when tumor suppressor proteins go to the nucleus of a normal cell, the cell just kind of waits, but cancer cells die because the tumor suppressors act. XPOVIO entered the clinic in 2013. Clinical studies were ongoing. We'll get into some of the interesting side effects and difficulties in dosing, but here we are with 2 FDA approvals already, another one on the horizon, we hope, and a couple more in the making with ongoing phase 3 trials. So this is a drug to address patients who have multiple different kinds of cancers. We're approved in myeloma, lymphoma, and more to come.
Great. Dr. Kaufman, thank you very much for your response there. Greatly appreciate it. And Dr. Huizinga, I'd like to welcome you to the camera. Thank you so much for your time today. Same question to you, and just as a reminder to yourself and the audience on the question, why did your company choose to focus on the disease state and indication associated with the product? In what ways did the product represent a therapeutic breakthrough? And how significant was the level of unmet medical need? Dr. Huizinga, welcome. And again, if you could remind our audience of your product and your background. Thank you.
Sure. Thank you, Ted. Well, hello, everyone. I'm Mimi Huizinga. I'm head of U.S. Oncology Medical for Novartis. So that means I oversee the medical affairs and clinical development within the U.S. for our oncology business unit. I'm here to talk about Dacvio, also known as crizanlizumab, which is a treatment for vaso-occlusive pain crisis in sickle cell. So there continues to be just a tremendous unmet need for treatment options for patients living with sickle cell disease. In the US alone, there are about 100,000 people living with the disease. Most of them are of African descent and ancestry. Sickle cell disease is a complex genetic blood disorder. The hallmark of the disease are unpredictable and very intense pain events or occlusive complications known as sickle cell pain crises. Physicians refer to them as vaso-occlusive crises, or you might have heard of VOCs. So historically, this high unmet need in managing VOCs, which can be associated with life-threatening complications, and are the main reason that people living with sickle cell disease go to the emergency room for treatment and are admitted to the hospital. And in the US, there are approximately 200,000 ER visits annually in the US alone for sickle cell disease and for the associated pain crises that I'm talking about. There's been very little innovation in sickle cell disease, and particularly around how to manage VOCs. There's this lack of treatment options is due in large part to the disease's complexity and how it affects the patient's body, beyond just how we think of and we're taught in medical school of the traditional sickling of the red blood cells and anemia. There are blood vessels and certain blood cells that can become stickier, which creates this proneness to clustering within the bloodstream. So it's not just the sickling, it's also the stickiness that impacts these cells. And when the clusters get large enough, this is when we can see the VOCs in patients, as well as other unpredictable complications. And despite the availability of a standard of care for many years in hydroxyurea, many patients on these treatments continue to experience pain crises, so they're not able to fully control their disease. At Novartis, we've been pursuing advancement to help patients living with sickle cell disease for many years, and we're very excited about our work with the Dacvio. It's the first FDA-approved, and it's a drug for sickle cell disease in many years. It's a once-monthly medicine that helps reduce how often these vaso-occlusive pain crises happen in patients, and it's approved for patients 16 years and older who have sickle cell disease. It's something that we can talk a little bit more about as we're going through the additional questions about how Dacvio helps patients and then what might make it the right fit for various patients.
Great. Thanks, Dr. Huizinga. That was great. And actually, just along those lines, if I can continue with you on our question
Absolutely.
number 2, I think it would be a great segue. So Dr. Huizinga, our question 2 is as follows. Is there a measurable impact you can cite on clinical outcomes for patients with this product? What's been the clinical response to the product thus far? Thank you.
Sure, yeah, this is a great question. So as sickle cell disease has been underserved with limited treatment options, we are hearing enthusiasm from physicians and appreciation for having a Dacvio as an option for their patients. We're excited about the wave of innovation that's been sparked around the approval of Dacvio in the last year, and are excited to see other products come to the market as well to help these patients who've been underserved for so many years. Dacvio is a prescription medicine approved to help reduce how often vaso-occlusive pain crises happen in these patients who are 16 years and older. And it's a monthly medication, and we found that it reduced the annual rate of sickle cell pain crises to almost half, so from almost 3 to 1.6 within a given year. And it also reduced the annual days of hospitalization from 6.8 to 4. So a 42% reduction based on the 1-year sustained study when it was compared to placebo. It works regardless of the sickle cell disease genotype and whether or not the patient is using hydroxyurea. And I think this is really important because, as I mentioned, hydroxyurea doesn't fully eliminate VOCs from patients, and it's not appropriate for everyone to use. And so being able to have this other treatment option, I think, is where the physicians have become appreciative that this is available and to offer to their patients. We did see that 36% of patients treated with the Dacvio didn't experience a VOC throughout the study duration in SUSTAIN, and that was compared to 17% of those who received placebo. I wanted, now that I've talked a little bit about some of the benefits, I also want to mention that there are potential risks with the Dacvio treatment, and it's really important that patients and caregivers speak to their physician about whether or not the risk benefit makes sense for them. The Dacvio can cause serious side effects, including infusion reactions. These might be signs of fever or chills or nausea and so on. And the most common side effects included nausea, joint pain, back pain, and fever. So it's really important that patients and caregivers speak with their healthcare providers about whether or not it makes sense for them to think about a Dacvio as part of their treatment plan. And we've heard continued positive feedback as the Dacvio has been commercially available. And we've heard from physicians that they view that the sustained data on the VOC reductions is clinically meaningful for patients. So, often in these clinical trials, we talk a lot about statistical significance, but I think we're really very mindful about what is clinically meaningful for patients. And so being able to reduce the number of VOCs that patients have per year or the time until their next VOC is really critical for these patients. And in fact, during the COVID-19 period, we didn't see any impact or any decrease to HCP administered treatments. So it's an infusion, so this means that patients have to go in. And we've seen patients continue to receive a Dacvio and physicians continue to prescribe it. And in fact, in April, the Sickle Cell Disease Association of America released their COVID-19 guidance, which encouraged patients to adhere closely to their treatment plans. And so we think it's really important for patients in this time of the COVID-19 pandemic that sickle cell patients continue to receive the treatments they need to avoid the pain crises and potentially reduce the need for hospitalization as well.
Thank you very much, Dr. Huizinga. If I could welcome back to the camera Dr. Kaufman as well. Dr. Kaufman, same question to you, sir. Is there a measurable impact you can cite on clinical outcomes for patients with this product? What's been the clinical response to the product thus far? Dr. Kaufman, if you could also just remind us the product you'll be discussing, I'll pass to you. Thank you.
Sure. Well, we're talking about Xpovio, oral selinexor, which is approved for patients with refractory multiple myeloma and also for patients with relapsed or refractory diffuse large B-cell lymphoma. The approval for multiple myeloma came about 1.5 years ago. And the study that it was based on was an accelerated approval, which means it was a single arm study based on response rate. Approximately a quarter of the patients, 25% of the patients had a bona fide response, which in myeloma means more than a 50% reduction in their tumor. Interestingly, 2 of the patients actually had undetectable tumor from this single agent, even though their disease had progressed on essentially every other drug that's available to treat myeloma. Now, myeloma is a disease with multiple different therapeutic options. 20 years ago, the survival in myeloma was 2 to 3 years. Today, it's overall about 7 years. But today, for the first time, there are patients living 20 plus years with this type of cancer because of all these new products that we have. Our product is approved after what's available. And because it's a single arm trial, it was tough to make direct comparisons. With good timing, though, we've now had two publications of Xpovio outcomes as compared to, quote, real world data, meaning patients treated at similar institutions with matched prior therapies. And the overall survival in our patients is approximately double that of patients who received other therapies, meaning recycling what their disease had already been treated with. And these were all these studies were done in academic medical centers, so very similar types of treatment paradigms. This kind of outcome in a single arm trial is, although it's not as good as a randomized trial, this real world data comparisons are being increasingly used. And we and frankly, clinicians believe that they are about as good as you're going to get in late stage cancer. It's not ethical to do studies in late stage cancer against placebo, especially in diseases where we know the inevitable outcomes, unfortunately, are going to be death. That all said, I will also provide that any patient with late stage cancer needs to be monitored closely and should be monitored, especially with late stage myeloma, particularly after a median of 7 or 8 prior regimens, which is about 15 different drugs. And patients should be seeing their physician regularly on a weekly or biweekly basis and monitored for the main side effects of the drug, which include nausea, low appetite, low platelet counts, and fatigue. Luckily, all of these side effects are generally preventable or treatable and are reversible in most patients. So we're very excited about the outcomes in myeloma. I would add that we have a pending approval in front of the FDA now in second line myeloma treatment, where Xpovio was combined with standard of care Velcade, which has been used now for 20 years. against standard Velcade. This was a randomized phase 3 trial. And in this trial, we did show definitive proof, at least as far as the efficacy outcomes are concerned, that the progression-free survival was significantly improved on the selinexor Velcade Dex or Xpovio Velcade Dex arm as compared to the Velcade Dex alone arm. In that trial, the use of Velcade and Dex was standard on the control arm, but I should mention is 40% lower on the experimental arm. So less of the available drug, a new oral therapy once a week as compared to standard of care. Overall, we think that these outcomes bode very well and should improve the outcomes of patients with multiple myeloma, both second-line myeloma and where we're approved now in refractory disease. Thanks.
Dr. Kaufman, thank you very much. Dr. Tu, if I could welcome you back to the camera as well. Same question to you, sir. Is there a measurable impact you can cite on clinical outcomes for patients with this product? What's been the clinical response to the product thus far? And Dr. Tu, if you could remind everyone of your product you'll be speaking to. Thank you.
Thank you so much. So again, I'm Ramon Tu and primarily focused on gilteritinib, also known as Zospata, which is currently approved in the relapse refractory FLT3 mutated acute myeloid leukemia. So as I mentioned earlier, one of the key things about acute myeloid leukemia is the poor outcomes associated with patients, particularly those with FLT3 mutations. And I think it's important to emphasize that after a patient had failed, there are papers that cites that the outcomes in terms of overall survival could be as poor as less than 6 months after they have relapsed at that point. So where does gilteritinib fit into the picture in terms of its impact for patients? Gilteritinib is a targeted therapy, and its initial approval by the FDA was based on an interim analysis that came from complete remission and partial hematologic recovery, also known as CR/CRh rates. And in this particular therapy, we were also able to demonstrate an improvement in overall survival in these patients. So that is 9.3 months for patients who received gilteritinib compared to only 5.6 months for patients who received salvage chemotherapy. This is a big change specifically for this specific disease. I remember this vividly as this is a disease very close to my heart, something that I have treated personally when I was a fellow in hematology and oncology, and even as a staff physician. And to see these patients go to your clinic and have limited response duration, and also having been requiring transfusions in a lot of times, and again shortened survival. So to actually see Zospata make a big change in these patients have made a huge impact. More importantly, this particular therapy is also an orally bioavailable agent. So many a times for patients who are diagnosed with acute myeloid leukemia, most of these patients would have to be admitted in the hospital and receive intensive salvage chemotherapy. So for Zospata, it's basically a monotherapy of an orally bioavailable agent that certainly helps improve the quality of life for these patients as well. It's also important to take note that those patients who received gilteritinib we were able to convert them from their transfusion dependence to transfusion independence of as much as 31.1%. So there's definitely been a big change in the specific area of outcomes specific to acute myeloid leukemia. From a disease where there's shortened survival, we've definitely made an impact specific to that disease.
Thank you very much for that response, Dr. Tu. And Dr. Tu, before we have you jump back, I have one more question for you, if you don't mind. It starts our third question set. Dr. Tu, what challenges did your company confront in transitioning from early proof of concept to human trials and ultimately to a market authorization? Any lessons from your experience that might add to a better public understanding or awareness of the medicine innovation process?
Yeah, thank you so much for that question. And again, going back to gilteritinib as mentioned, I think as Dr. Kaufman and Dr. Huizinga has mentioned earlier, I think all of our companies are very dedicated to advancing the cause for patients, not just in hematologic cancers, but even in diseases like sickle cell disease. And it's very important that we continue this specific tradition. But nonetheless, many a times, people only see the product when it's done, but there's always a story behind these compounds and how hard each of the teams, the patients, their caregivers, and their family members have sacrificed to make this happen. So gilteritinib is not an exception to those challenges, of course. The initial challenge was to select the right indication for the compound. So ASP2215 was initially identified only as a backup candidate for another molecule, and it was intended as an ELM4 ALK inhibitor, which was being developed for ALK positive non-small cell lung cancer. And based on its target inhibition profile, ASP2215, which is what we call gilteritinib, also could have been developed for lung cancer and other indications. Thankfully, we were able to see this through. We know its specific target profile and its ability to inhibit FLT3. So we focused our attention specifically for FLT3 mutated AML, and we were very fortunate there. So I think the key take-home message there is to somehow understand your target profile for a specific molecule and hone in on that specific target as indicated. The other important thing, and this holds true even for diseases that Dr. Huizinga and Dr. Kaufman had mentioned, and that is many of the diseases that we're talking about at this time are diseases that are not that common. And so the patient population is very critical. So enrollment for the specific studies has been a challenge, thankfully, again, through the commitment of the company and also the investigators. And of course, the patients who have basically taken, decided that this is a specific trial that they would go for, have also been committed to that. And I think an important lesson also specifically to this compound is we should always strive to do things faster. And the best way to do things faster, because our patients need them, is to always challenge the norm. And that is to consider non-traditional study designs. One of the key things that gilteritinib did that allowed it to move forward to where it's at is that it used an accelerated titration design and also allowed us to expand in specific dosing levels where we've seen initial clinical activity. So I think these are the key aspects of the study that hopefully can be taken on as well by other groups later on who are interested in advancing the compounds. Thank you.
Dr. Tu, thank you very much for your response. Dr. Kaufman, if I could welcome you back to the camera. Same question to you, sir, on your product. What challenges did your company confront in transitioning from early proof of concept to human trials and ultimately to a market authorization? Any lessons from your experience that might add to a better public understanding or awareness of the medicine innovation process? Dr. Kaufman, thank you.
Sure, thanks. I think when you come up with a new medicine, you have to be ready for a marathon that is going to be maybe even a double marathon. I think that's the first thing, and you've got to believe in what you're doing and that you can get to the end. Coming in, finishing 20 miles or 30 kilometers on the marathon is not sufficient. You've got to finish the race, and you want to come in with being a winner, at least in the top 3. So you've got to be ready for that. We embarked as a small company with essentially angel funding and funding from those of us that founded the company, really starting in Dr. Shackham's home kitchen and home computer with an idea. And we're fortunate enough to run into someone who funded the Series A financing with a completely new idea based on the people involved. And new ideas are a dime a dozen. Great ideas are only true after they come to fruition. So luckily, this has been a good idea. We first put this drug into animals, and one of the main side effects is weight loss, which has a whole bunch of other positive potential, but that's a separate discussion. But we were looking to see that the cancer shrunk in the animals, and particularly in mice, without the mice really getting terribly sick, and luckily they didn't. We then actually moved into dogs because dogs get spontaneous cancers, and they turn out to be decent models for human spontaneous cancers. And many pet owners want to treat their dogs. So one thing we learned early on is mouse cancer has been cured many times, and what happens in mice doesn't often happen in humans. So you really have to try for new models. Dogs represent a potential new model and have been used with drugs like Imbruvica and with our drug, and potentially will be positive. and be approved actually for pet therapy, for dog therapy with lymphomas, with our a compound related to the one that we have approved for humans. We then moved into monkey studies to make sure the drug was safe. So the drug was safe in mice and rats and dogs. And actually the precursor to the compound that was eventually approved in humans caused a lot of side effects, neurological side effects in monkeys. And this was not seen in any of the other animals. So while everybody wants to reduce our reliance on animal testing for safety, here's a case where only the primates got into trouble. And thankfully that drug never made it into humans, because almost for sure we would have seen trouble in humans. That led to the testing of a second compound in monkeys, which showed good tolerability. And that's selinexor Xpovio, which eventually went into humans in 2012. As we ran through the early development process, even in humans, this being a new therapy, it's an oral therapy, and it doesn't cause major side effects. It causes annoying side effects that especially patients with late stage cancer just don't want to put up with: nausea, fatigue, reduced appetite, change in taste, and things like that. Unfortunately, in cancer, we tend to try to power through these things and dose more and more drug. And that for this drug, that was the wrong strategy. So we ran into this strategy where the drug got really good and then started to wane as we kept going up on the dose. And we had to learn that actually middle doses were better than higher doses of this drug. Side effects were less and efficacy was as good or better at the middle doses. And those are the doses that are approved in humans. And subsequently, we ran into some bad timing on the regulatory front, where another drug that was being developed in myeloma had a very difficult phase 3 outcome. Things looked pretty good on some of the efficacy parameters, but the survival was negative for that drug. And our timing was such that the FDA sort of put a screeching halt on all myeloma drugs at that moment. And we got stuck in a difficult advisory committee where we had a negative vote, 8-5 against us. But FDA actually saw through that and listened very carefully to the commentary and the experts at the advisory committee, took a look at our early randomized data, even data that we weren't allowed to see, and made a decision to, quote, give us a chance. They were convinced by the early randomized data that we likely had a good effect, and eventually we got the approval. But this was brutal. For a small company with minimal funding, we savvied through that. We got through it, and FDA gave us the approval, and the outcomes have been very good now that the drug has been on the market, as I discussed before. So this is not for the faint of heart. The last thing I would say is that everyone should realize, and we need to talk about this more as pharmaceutical people, the cost per patient for trials is higher than $100,000 per patient to test this drug before it ever sees a regulatory submission. When we submitted this drug for approval, we had treated over 3,000 patients, costing us, just for the patient costs, not for employees, over $300 million for this drug to get its first approval. And I think we need to be honest about this. If we want to continue to develop safe and effective therapies, everybody needs to recognize the hundreds of millions and sometimes billions of dollars that are required to get us to the finish line and make this drug generally available now, and give patients another shot at more milestones in their lives.
Dr. Kaufman, thank you very much for your response there. Greatly appreciate it. Dr. Huizinga, I'd like to welcome you back to the camera, and same question for you, please. And that question, again, as a reminder, is what challenges did your company confront in transitioning from early proof of concept to human trials and ultimately to a market authorization? Any lessons from your experience that might add to a better public understanding or awareness of the product innovation process? And Dr. Huizinga, just if you could remind us your product again, since we've gone back and forth a little bit of times. Thank you and welcome.
Sure, thank you. And I will try to build upon what Dr. Tiao and Kaufman have said and not try to repeat, because they made several good points. So as a reminder, I'm talking about crizanlizumab, also known as Dacvio, which is a treatment for the prevention of vaso-occlusive pain crises and sickle cell disease. I wanted to come back to one of the initial points made around, the thinking about how patients can participate in the trials and how, indebted we are to patients that do participate in our trials, and how we can try to think about ways to be more patient-centric in our approaches. We had a lot of socioeconomic and logistical access barriers for patient participation in all clinical trials. And that was not, this trial that we did called SUSTAIN and sickle cell disease was no exception. We did try to do some innovative things with this trial to make it more accessible for patients. We put in place a patient support program that leveraged a debit card to have trial participants be able to access the money that they needed for study-specific visits. So that could include ground transportation from and to home and the clinic through a car service, or reimbursement of lodging and transportation expenses that were specific to the study and to the needs that they had around it. In addition, one of the key challenges in sickle cell disease is just a distrust from the patient community, especially those of African ancestry, with the medical and healthcare industry in general. And there were fears of exploitation in the medical research, given historical practices that have tarnished the relationships with these populations. So at Novartis, we always have a patient-centric approach in our research and development, which allows us to incorporate the patient voice in the trial design process. And in this case, we wanted that same patient voice to help us not only with the trial design, but to think about some of these key participation barriers. And we wanted to partner and seek to understand. We did a lot of listening through the design of these trials, and then also in how we built strong partnership with several of the patient advocacy groups. So by sharing their perspectives around the research design, the patients weighed in on many factors. So everything from the primary and secondary endpoints for the clinical trials. So they help us understand what might be clinically meaningful for patients with sickle cell disease. The patients also helped us develop the patient-reported outcomes. So PROs are pros for many of us in the research world. So we had specific instruments developed for sickle cell disease. Patients informed our trial protocols, the informed consent documents, and all of this to ensure that the drug being developed would indeed provide some value of significance to patients, particularly those in underrepresented communities from prior trials. So we've been in the sickle cell space for many years, and we're committed to staying in for many more. So we've worked hard to build strong partnerships with several patient advocacy groups. And these groups, I feel like this was mentioned in some of the others, they're just really critical for partnering with us in order for us to make sure our trials are designed appropriately, and then to make sure that we're sharing important information and strengthening our ties with the patient community in general to make sure that we're bringing back information. And we've worked with many, so Sickle Cell Disease Association of America, Sickle Cell 101, Sick Cells, Sickle Cell Community Consortium, and Bold Lips for Sickle Cell. And then worldwide, we also worked with the Global Alliance for Sickle Cell Disease Organizations, which have many different patient advocacy groups that are associated with it. And I think that going forward in the future, this type of commitment to patient-centered research is really important. And it's important for us to find ways to work with the patient community, either through existing groups or to create the space for groups where there might be, in some of the cases that we talked about, these are rare diseases. So how can we find existing groups or create space for the community to rise up and to help us create meaningful trials, and then to also address potential barriers to participation in these trials.
Dr. Huizinga, thank you very much for your response. And to all of our panelists, thank you so much for your input and responses to my questions today. Really, really great innovation taking place, and exciting to see all the updates taking place in your respective therapeutic areas. Regarding innovation, I'd like to end our discussion today with one final topic. And that topic is regarding innovations trending in your individual product area. Dr. Tu, if I can come back to you for starting us off on this final topic. Here are my questions that I hope you would address and give the audience answers to. As scientists and clinicians, where do you see the next wave of innovations trending in your individual product area? What new areas of medicine and the fight against these conditions excite you the most? And what kind of innovations might the Galien Awards Committee be reviewing in your field 5 years from now? Dr. Tu, thank you, and I'll pass to you.
Yeah, thank you so much, Theodore. So going back, so I'm primarily focused on gilteritinib in terms of the discussion that I had earlier. And in terms of innovation that's coming through in this specific field, so within AML, drugs like gilteritinib have paved the way for targeted treatments against specific molecular mutations that tumors depend on. And in our case, this is FLT3 mutations. And as we move forward in the next 5 years, drugs that target other hard to treat molecular subtypes of AML might come to light. And I think this is a very important area, as there's still a lot of specific subtypes of AML, specifically those with molecular subtypes that we don't have effective therapies for. Investigations of drugs that target the leukemia stem cells are also underway, and this could potentially lead to more long-lasting outcomes. So as many of you are aware, specific leukemia stem cells are very important to target, as many of these cancers can easily relapse after initially responding to therapy, because it's believed that these leukemia stem cells are not necessarily adequately treated with currently available therapies. And as cancers like AML depend on multiple mechanisms to survive, combination therapy with other drugs that target different tumor dependency mechanisms will be very critical. And of course, there's also the field of immuno-oncology, also called IO. And this has been the buzzword in the field of oncology for several years now. It has definitely revolutionized the treatment in many cancers, including solid tumors, cancers like melanoma, lung cancer. This is a very broad field and includes classes like cell-based therapies, immunomodulators, vaccines, antibody-based therapies, and oncolytic viruses, just to name a few. And in cell-based therapies like CAR-T, having already shown some outstanding results in certain types of blood cancers like ALL and certain types of lymphoma, the hope is that these therapies can also deliver similar efficacy in AML patients.
Dr. Tu, thank you so much for your participation today and for all of your responses. We're truly excited to see the continued innovation from both yourself and your company, and look forward to seeing you again soon. So thank you very much for your time. And Dr. Kaufman, if I could come back to you, sir, again for the last question and topic. Again, as a reminder, the question is as follows. As scientists and clinicians, where do you see the next wave of innovations trending in your individual product area? What new areas of science in the fight against these conditions excite you the most? And what kind of innovation might the Galien Awards Committee be reviewing in your field 5 years from now? Dr. Kaufman?
Yeah, one of the challenges now that the war on cancer is being fought on hundreds of fronts is to figure out how to combine these fronts and optimize the combinations of therapies that we have, and also the timing of these therapies. You know, the way we traditionally do things with cancer is we start with patients who've been through the available treatments, and we give new drugs to those patients. And if we see something, we start to move up in the paradigm. We're no different. This cannot continue to be efficient, because there are probably some very important therapies that would be really effective early on, perhaps targeting stem cells and the like, that would be a lot less effective and maybe not even effective at all later on. So we're missing them. The second problem that we have is how to combine these most efficiently. And this is where systems biology and some of the artificial intelligence start to come in. Systems biology is a bit of a misnomer, because frankly, all of biology is a system. But because we study things biochemically in a reductionist way, we change one thing in the laboratory, we find an effect, and we go back. I never met a patient who started with one pathway. You know, patients come in with all their medical history in addition to their disease history and so on. So without being too facetious about it, we need to come up with better systems biological ways to combine drugs, and also to target when to use those drugs based not only on the tumor, but as we're increasingly finding out, and Dr. Tu mentioned, based on the patient's own immune system and their other physiological characteristics that are going on. Lots to do, and we're starting to break into that area, but there's a great deal of work to be done in all of those areas. So I'll leave it at that.
Thank you very much, Dr. Kaufman. Thank you for your time today. Thank you for your entrepreneurship and taking that and applying it to healthcare and looking to benefit patients. And we truly are excited for the innovation that you and your company will continue to bring. So thank you very much. And Dr. Huizinga, if I can welcome you one last time as well too. Thanks so much for your time today. The final question again to you. As scientists and clinicians, where do you see the next wave of innovations trending in your individual product area? What new areas of science in the fight against these conditions excite you the most? And what kind of innovations might the Galien Awards Committee be reviewing in your field 5 years from now? Dr. Huizinga?
Great, thank you, Ted. Well, Dr. Tu and Dr. Kaufman were able to speak about oncology in general, and I think being able to focus in and talk just about sickle cell disease, I would say it's hard to mention sickle cell without thinking about the potential for CRISPR. I'm sure all of us have heard about the gene editing and the gene therapy that we might be able to offer patients with sickle cell, and again, striving for one day when maybe there's a cure from this terrible condition. That said, gene therapies take a very long time to develop, to come into play, and I don't think that they're going to be appropriate for all patients. So given that, Novartis remains committed to the broad space. So in addition to our early and exploratory efforts in CRISPR, we're looking at a number of other pathways within sickle cell, and we're not finished with crizanlizumab yet. We'd like to understand how crizanlizumab might benefit patients younger than the age of 16. We also have trials looking at how crizanlizumab might affect some of the other complications that these patients have. As you know, that many of these patients with the vaso-occlusive crisis and the block of blood flow, they have end organ damage, and almost every organ in the body can be impacted. So we would like to understand more about how we can prevent some of those complications that happen. We call them the end organ complications from sickle cell disease. And we see that not only crizanlizumab or Dacvio might play a role in that, as well as some of the other mechanisms that we're looking at within the sickle cell. So we're very committed to sickle cell, and playing off of my kind of other answer, it's the excitement around what we're doing in the disease area, but also how we're trying to partner with patients, and then how we're trying to work and expand our clinical trials into sub-Saharan Africa, and to make sure that we're thinking about the patients who would benefit from these types of therapies, and that we're giving a chance to reduce the barriers and include them in the clinical trials that we're planning.
Dr. Huizinga, thank you very much for your response. It was such a pleasure to spend actually 2 hours with you today on 2 different panels. So thank you for that opportunity, for joining us, and we're truly excited for the innovation that you and your company will continue to bring to healthcare. So thank you very much. And to our audience, thank you so much for your time, as we heard some great responses and input really truly on innovation in this area of medicine. At this time, we also want to thank you for participating in our poll at the beginning of our series, our lecture today. At this time, if I could ask the Galien Foundation to please post those results, and thank you. Everyone should begin to see that on their screen. I'll just review the results fairly quickly. Our question today was, what is the most important single element of a future global policy agenda to advance health innovations that improve the human condition? 40% responded universal access to medicines and vaccines. 30% responded investment in population-based primary care, prevention, and the social determinants of health. 10% response on empowerment of women, reproductive planning, and expanded education for young people. 10% response on multilateral collaboration and health policy, including return to US participation in the WHO and other global, and 10% response on more public trust in science. Again, thank you for your participation. We will be gathering all the results from all of our panel discussion today, and that will be presented later on. So thank you again for doing that. And with that, I just want to wrap up by thanking all of our company representative and our panelists. Thank you so much for your time today. Specifically, we thank you for your time, we thank you for your service, and we thank you for your dedication to both medicine and to healthcare. So thank you so much for participating with us today. And to our audience, we know how valuable your time is. Thank you for joining us today. Please note that this webcast is viewable on the Galien Foundation's homepage. So please visit us at the Galien Foundation's homepage to see this webcast, as long as other webcasts that took place both earlier today and later on. And finally, we would like to note Thursday's forum, the awards cocktail and presentation of the 2020 Vagelis Pro Bono Humanum Award winner, Dr. William Fagey. And with that, again, thank you so much for joining us today. Thank you again to our panelists. We hope you have a great day, and we'll hope you join us for future sessions. Thank you.
Best pharmaceutical product.